This video was produced in the framework of the Climed-Fruit project, and will allow you to discover the GASCOGN'INNOV Operational Group and the different results and testimonies that resulted from it. Starting, a little tutorial to remind you how to put YouTube subtitles in your language. First, click on the Settings button. And on the Subtitles button. You can click on the Auto-Generated Proposal. Once it is done, click again on the Subtitles button. Now choose your language. Here we are, now you are ready. Let's start with the general presentation of this operational group.
So the GASCOGN'INNOV project was a project funded by the European Partnership for Innovation for 5 years, from 2017 to 2022, and it brings together wine grower, researcher, advisor, and wine technician to answer to a question on how to improve soil quality. So the background of the project was a project called GASCOGN'INNOV, who was a project of collaboration of researcher and farmers in several crops, including viticulture, to validate a set of analytical indicators of the biological quality of soils. And this project also developed training on soil biology and a transfer procedure. In GASCOGN'INNOV, we adapt these indicators for viticulture in two objectives: acquire regional reference on the impact of viticultural practices on the biological functioning of the soil, and the second one on crop management, in the idea to co-construct crop itineraries that meet the challenge of biological quality. To answer the question, is my practice is good or bad for soil quality? So the project was facilitated by EFV with 18 partners. So 15 were wine estate with a good repartition between cooperatives and independent wine growers. These wine growers were supported by some advice for education services as EFV, but also as agricultural chambers and advice on biological viticulture. And the project, the wine estate in the project were between 15 to 300 hectares. And 6 external partners, scientific partners, were helping the OG to decide on the best itineraries to protect soil quality. So the set of indicators of biological quality was evaluated in the project. The network was constructed of 13 plots. One plot is one vine plot. In 2016, we constituted the network. In 2017, we conducted an initial assessment, so using the indicator from GASCOGN'INNOV to make a diagnosis on soil quality and characterize the crop system. So the diagnosis permitted to identify several agronomical practices to restore soil fertility. For example, plant cover, organic matter inputs, reduction of herbicides, mineral fertilizer, and some practices were experimented in the field with an agronomic evaluation. So here's the co-design with the experts. We identify action plans. Then the actions were implemented on the plot with an agronomic evaluation. And in 2021, we made a final assessment using the same indicator as the first one to characterize the system and to see how it was improved. And last year, we made a restitution to the group. So the particularity of this OJ is that it's based on a participatory approach. So first step was a training. EFV and the biological experts were training the wine growers and the advisor to use the indicator set. That means how to measure each indicator or how to make some samples. Then each wine growers perform the picking of samples by themselves or with the support of advisory structure in the following months. Then we analyze the results with the external group of experts. Each expert has a specialty on each compartment of the soil. So there is one for the microorganism, one other for the macroorganism, and so on. So system redesign was made in collaboration by wine growers and the interdisciplinary groups to identify practices to implement on the plot. And then during the restitution phase. And it led to action plan of co-conception. So that's all for the structure and the methodology of this OJ.
Let's continue with the testimonies of some members of the project.
Donc je me présente, François Dargelos, je suis viticulteur dans le Gers en Gascogne, voilà, en appellation IGP Gascogne. Et mon implication dans le projet GASCOGN'INNOV était en tant que parcelle test de pratiques innovantes que l'on avait choisi de mettre en place. Sur mon exploitation, composée majoritairement de cépages blancs, voilà, pour une surface d'une quarantaine d'hectares. Voilà, je suis installé depuis 1995. Voilà, sur un vignoble plutôt vigne large, avec des encépagements blancs sur limon sableux. Voilà à peu près les typicités de mon exploitation. Alors, si je fais un bilan de ce suivi sur les années passées, c'est me permettre, en fait, ces indicateurs me permettent de prendre un peu du recul sur mes pratiques, d'avoir une vision un peu plus globale, d'avoir acquéri des connaissances en côtoyant un peu les scientifiques et les chercheurs. Et au final, peut-être d'arriver à orienter un peu plus ma façon de travailler voilà, sur le terrain, avec des couverts végétaux, avec des apports exogènes, de façon à ce que je sois moins agressif et que je rentre dans un système peut-être un peu plus vertueux, plus résilient, voilà, qui s'inscrit dans le temps et qui répond aux changements de demain. Alors, la démarche collaborative, elle a amené une construction collective du projet avec des échanges qui nous ont fait prendre du recul, qui m'a fait prendre du recul, de par parfois les questions du groupe ou les interrogations, voilà, ou les projets proposés par les autres qui ont fait que ça faisait sens aussi chez soi. Donc oui, la démarche collective, elle a été hyper intéressante, même si après, l'objectif était d'appréhender chez soi comment ça fonctionne et mettre en place des choses chez soi dans son contexte.
Donc, je suis Carla Fabry, technicienne à la Chambre d'agriculture du Gers. Et donc dans GASCOGN'INNOV, je me suis occupée de la partie technique terrain, d'accompagner les viticulteurs dans les prélèvements destinés aux analyses pour GASCOGN'INNOV. Les projets comme GASCOGN'INNOV permettent de créer une dynamique et une inertie au sein des appellations et du vignoble gersois, et derrière de pouvoir créer des journées de restitution qui également contribuent à créer une cohésion pour les viticulteurs gersois et les techniciens.
Now that you know more about GASCOGN'INNOV, let's see the results that came out of this project.
Okay. So the main deliverable of this project was a set of indicators to evaluate soil biological quality. This indicator include 3 bioindicators: the microorganism, fauna, and earthworm abundance and diversity. Also, it includes an indicator on the structure and the physical chemistry of the soil, which we call the lodge. And the last one is the food. It's an indicator of the degradation rates of organic matter, which is called leva bag. It's a small bag with straw inside that we put into the soil, and we wait until it disappears. So in all the plots studied by the project, 2 types of practices were implemented and tested to improve soil quality: introduction of green manure or organic inputs. So as a result, we made a characterization of the technical itineraries of the sampled area with several indicators, some qualitative indicators as the frequency of organic amendments, the restitution of pruning woods, the yield, fertilization, herbicides, and quantitative indicators, the duration of the vegetation coverage of the soil, and the tillage intensity, number of passage, type of tool, and depth of work. So this approach allows us to determine 3 groups of plots. So group 1 and group A and C have the same soil characteristic, but group A has extensive mechanical tillage and less long and less diversified vegetation cover, whereas group C has little mechanical tillage and long and diversified vegetation cover, and the group B is a different type of soil with moderate mechanical tillage and medium vegetation cover. So what can we say of that is that the soil type has the main impact on the biological parameters. We found also an effect of soil management practices. So especially decreasing the intensity of tillage and increasing the duration, the time of presence of vegetation cover tended to, and diversity, tended to increase the abundance of all the organisms studied. So more vegetation and less work, to be very simple. So one example of practices tested in this project, so it's implementation of green manure, some results of green manure on the quality of the soil. So in this test, we introduced Vicia faba bean, Vicia faba. It was a clay and limon stall, and the objective yield of the plot was high because it was EGP production. This plot was followed from many years. So we saw a positive effect of green manure in the organic matter. So it's what you have in green. Green, it's the green manure, and in yellow, it's the control. And the control is labor of the soil. Green manure has also a positive effect of microbiological biomass with a high increase, and around 2,000% of increase in earthworm abundance, and also a good earthworm diversity.
Ouais.
Diversity. Yeah. But as we are agronomists, we also investigated the effect of green manure on the yield and on the nitrogen content. So in this plot that we followed from 2015, we saw that the green manure keeps a high yield. And even if this plot has not, the objective of the green manure in this plot was not to improve the yield, it was really to improve the soil quality and diversity. But we saw that it has also a positive effect on yield with a slight increase of yield starting in 2018. The same for nitrogen content in berries. It's the green manure also allowed to improve nitrogen content in berries. That is a good point for an easiest beneficiation.
To put things into perspective, here are the strengths, weaknesses, opportunities, and threats analysis that were conducted on the example previously presented.
So in conclusion, which practices to boost soil quality? So in the project, many were tested, but the more easy to transfer is the green manure. We saw that the importance of plant cover, maximizing the time they were present. It's a very good point to improve soil quality, but it's also a global reasoning. Some practices can compensate the deleterious effect of others. That means that you can still have labor of the soil, but in other time of the year, you can compensate it by using cover crop. And improving soil health takes time. It's very slow. This project was 5 years, so it was good, but it takes time. What is the place of soil biological quality indicators? To answer the question, is my cropping system improving or degrading the quality of my soil? So this type of indicator can be used to answer this question and to help to design itinerary more respectful of soil quality. And it could be good to monitor this biological indicator every 3 to 5 years to control the sustainability of practices.